Hello, everyone, and welcome to today's webinar, The Power of Autofluorescence Extraction Using Full Spectrum Profiling. I am Laurent Ginestidaraki, Product Manager here at Cytek Biosciences, and I'll be your moderator for today's event. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We have about 10 minutes to answer questions at the end of the presentation live, and more information can also be found at www.cytekbio.com. Thank you to LabRoots for hosting this webinar. It is an educational webinar and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Diana Bonilla Escobar, our U.S. Technical Lead for Application Support here at Cytek. Diana, you may now begin your presentation.
Morning, everyone. Thanks you for joining us today. My goal is to show you the power of using full spectrum profiling when you are understanding the autofluorescence in cells in suspension by using flow cytometry. This is the first part of a 3-lecture series focused on this topic of autofluorescence, which is that natural emission of light that we observe in cells. And what we will discuss today is 3 main topics. First, autofluorescence. What is it? Where does it come from? How it changes? And then that particular technology that we use, full spectrum profiling, what it is about, how it works, and finally, how we can use that technology in order to understand autofluorescence, the main player for the talk, characterizing that autofluorescence, and finally, being able to extract it if needed in order to get better resolution for your flow cytometry data. If we start with autofluorescence in microscopy or in flow cytometry, we know this as background. It's noise, baseline signal that is just coming from unstained cells. All that natural emission of fluorescent light that's coming from biological structure within the cells in your sample. And the ability to capture autofluorescence has been used in multiple fields, including clinical applications, because changes in autofluorescence can also serve as diagnostic indicators. For example, this is an image of our right eye that shows hyper autofluorescence in a patient with vision loss caused by toxicity. And the point is this autofluorescence, hyper autofluorescence in the perimacular area was critical as an indicator for diagnosis. If we focus then on cells in suspension, on flow cytometry, or even microscopy, as the images I'm going to show, autofluorescence, it's all the light coming from cells that is detected in the absence of any exogenous stain or reported agent. What I'm showing you here, it's cells, leukocytes, different types, after they are excited with a UV laser. What we are seeing is natural emission of light in different patterns, different intensities, and those differences are going to be based on how big the cells are, how granular they are, also their metabolic state. For example, if we compare leukemic cells with normal cells, in the leukemics, we see that autofluorescence in a more uniform distribution. We barely see any of the cellular structures that you could see in a normal cell. And it's not only changes in how the light is looking, the distribution, the amount of light and the pattern, it's also the wavelength at which that light is being emitted. If we compare, for example, different wavelengths from 400 to 800, what we are seeing is that untreated cells, they have more of a blue emission profile versus cells treated with a cytotoxic agent, it's more of a red emission, what we could be capturing. So multiple differences in that autofluorescence, in the different parameters for that autofluorescence characterization, and how that changes based on cell size, granularity, the metabolic state, and different experimental conditions as we are seeing here. So autofluorescence comes from endogenous molecules that become fluorescent when they are excited by the lasers in your cytometers. That includes components involving structural or metabolic functions in the cells, structural proteins like the ones in the extracellular matrix, like collagen, elastin, for the organelles, all the dynamics in fusion, in clustering of these organelles in response to metabolic stress. It's going to be influencing autofluorescence. And in terms of metabolites, we have metabolic cofactors, amino acids, lipopigments that also emit autofluorescence. If we characterize that emission in terms of the wavelength of emission, we see that usually most of them, they absorb in the UV to blue range and ended up emitting that light from the blue to the red range as well. The different proteins have characteristic excitation emission profiles, and the different components also are related to specific cell types. If we want to understand autofluorescence, it's a very dynamic process. It can increase or decrease based on multiple factors. One of them is physiological processes. When cells are growing, when they are proliferating, when they are producing energy, there is higher autofluorescence. Also pathological conditions. We know stress response, oxidative damage, disease, things like hypoxia, inflammation, cancer can change the autofluorescence pattern. And in experimental conditions during the staining protocol, for example, we know that aldehyde fixation, transfection reagents, hybridization buffers increase the amount of autofluorescence in the cells. In general, what we know is that we will have higher autofluorescence levels in cells that are larger, that are more granular, cells that are stressed or dying since they are producing more energy and more proteins, and also cells with more content and more activity in lysosomes and mitochondria are going to be having more autofluorescence. We know then that autofluorescence for microscopy and for flow cytometry, it can be problematic because it can interfere, that baseline can interfere with the detection of ureal fluorescent light, masking low expressors or even interfering with dim fluorochromes or low express markers. What you can do with autofluorescence, the traditional approach is just work around it. Reduce that unwanted autofluorescence by using quenchers. This is an example. These are red blood cells. This is the real fluorescence. And what we are seeing is that if you quench the red blood cells, you ended up seeing the specific fluorescence you are interested in. Another tool could be optimizing your staining protocol, reducing the concentration of your fixative, reducing the amount of serum that is present in the staining buffer, removing dead cells, removing extracellular matrix, lysing your red blood cells could help as well. And finally, a way to work around autofluorescence is use red fluors. Fluors that are not going to be impacted by that light that autofluorescence it's adding into your samples. Things like APC, Alexa Fluor 647, Alexa Fluor 700 are not impacted, as we will be seeing in the plots, by autofluorescence. So a good option to characterize markers in that area. Another option, once you have characterized or captured the light from autofluorescence and the light from your real fluorescence signal, in this case GFP, what you can do is computational removal of that background in order to get better resolution, better signal to noise ratio. That's what we call extraction of autofluorescence. And finally, what you could do is take advantage of the signal you are capturing in autofluorescence or in unstained cells. This is an example. This is a transverse section of a pine needle. We are seeing here is after exciting the cells with a laser without any hexogenous dye or staining. What we are seeing is the natural emission. Chlorophyll, this red emission, and also flavonoids on the cell wall. So plants have pigments that are going to be fluorescent after excitation by multiple lasers, and that's what you could capture. So that's critical not only to take pretty pictures, but also to understand changes in that emission profile when you have photosynthesis, CO2 assimilation, changes in pH, or also treatment with different chemicals and measure those differences without using any label or staining. So full spectrum profiling, going back to the other player in the talk, full spectrum profiling is a technology that enables you to do either extraction, correct, characterize this autofluorescence and remove it by doing extraction, or use all the natural staining in your sample to answer any scientific question you might have. Let's talk then now about full spectrum profiling, what it is and how it works and how we can use it for autofluorescence characterization. Full spectrum profiling is a technology introduced by Cytek back in 2017, uses flow cytometry to characterize single cells, cells that are in suspension, cells that are passing through lasers, and lasers are illuminating those cells. Then light is going to be scattered, fluorescent light will be produced, and we will be collecting that light. Three characteristics of this full spectrum profiling are, number one, it gives you high resolution data because it has low noise electronics and high sensitive detectors of light. We use APDs or avalanche photodiodes for that determination. And the high resolution means that at the end, if you're interested in cells that are positive for one marker, cells that are negative for one marker, the separation between negatives and positives is much better because of the detectors we use and the low noise in the system. Since we part of the full spectrum profiling is doing a full characterization, full across multiple wavelengths in the visible range. So we build spectral signatures. We will talk more about that, how we do it. But the key message is that since we do that full characterization for samples, for fluorochromes, what we have at the end is the ability to put together more dyes in the same sample to characterize the same cells. What we have reached so far is that using a unit with 5 lasers, 64 detectors for fluorescence, we can go up to 40 colors, 40 different proteins measured in the same cell simultaneously in order to characterize things like immune responses. Finally, a great feature of using full spectrum profiling is the characterization of autofluorescence. Because similarly to what we could do with fluorochromes with multicolor samples, we can characterize unstained cells and understand the autofluorescence, and if needed, then extract it in order to have better resolution. How the system works. The system has multiple lasers. Here showing you a unit with 5 lasers, UV laser, violet, red, blue, and yellow-green. The lasers are specially separated. They reach the flow cell, the interrogation point where cells are going to be receiving that light from the lasers and then emitting light. All the light that it's emitted will be collected in multiple detectors. We have detectors like this SSC, side scatter detectors, that collect the light that indicates granularity in the cells. We have 2 of them, one after cells are excited with the violet laser and one more after blue excitation. We have one detector, forward scatter detector, that collects light that gives us an idea of how big the cells are, frontal light. And in addition to that, we have 64 detectors that collect fluorescent light after fluorochromes are excited, after fluorescent proteins are being excited as well. What we have is modules of detectors. For example, the UV module, it's going to have 16 detectors total, detectors that will collect light from shorter wavelengths around 380 to longer wavelengths around 800, characterizing all the emission after the cells are excited by the UV laser. So UV laser excites molecules, that light is collected by a particular module with multiple detectors in the module to capture the full range of emission. Similarly, we will do with all the other lasers and all the other modules. One dedicated module for the yellow-green, for the red, for the blue, and the violet laser. After capturing all that light in multiple detectors, what we build for the fluorescent light is a spectral signature. Spectral signature is going to show you on the y-axis the intensity of the signal, how many photons are captured by those detectors of light and then converted into electrons. We will also be seeing on this plot on the x-axis the detectors of light. This is the UV module. Detectors organize shorter wavelengths to longer wavelengths, covering the full range of potential emissions for a particular fluorochrome. From here, we can see what detector is capturing more light, more intensity, definitely UV14. And as I mentioned before, we will do the same for every excitation. After violet laser is exciting the cells, we capture the signal, blue, yellow, green, and red. And we build the full spectral signature, putting all that information together. in a single plot where we have all the detectors of light in the system and the intensity that each detector is capturing. From this, I can conclude, for example, this particular fluor, the highest emission is out of violet detector 14, and is excited by multiple lasers. This is the key difference between using full spectrum profiling and conventional cytometry. By conventional cytometry, instead of having multiple detectors, what you have is one single detector that captures the peak of emission for that fluorochrome, versus having spectral characterization where you have a more global understanding on how that fluorochrome is excited and it's emitting light across multiple detectors in the different modules in the system. What are the benefits of having that full spectrum characterization and having those specific detectors? It's the point that now we understand better the samples, understand better the fluorochromes, what's their pattern of excitation emission. Doing that and not only focusing on one single detector, we now can use dyes that have a similar peak of emission, but their whole signature is different, and we can use them in combination for the same panel and characterizing the same cells. And that happens for multiple fluor pairs. Since we can put more and more fluors, then we'll be able to build larger panels. The example is a 40-color panel that we recently published out of a single tube with high resolution because of the optical configuration and layout that I described. In addition to that, we can save sample by the fact that from a single tube, we can gather more information. If you're interested, you can scan and read the full story on how we went from 3 tubes for characterization using a conventional cytometer to only 1 tube, again, saving sample and doing a more global characterization. And finally, the benefit of full spectrum profiling is the fact that if you can understand autofluorescence in unstained cells, then you will be able to extract it in order to see the real fluorescence, in this case from a fluorescent protein, where it was difficult if the background is not extracted. We will focus now on that, how we do extraction. But before, let me remind you that if you're interested in full spectrum profiling, this is our website, cytekbio.com. In there, you will find a lot of resources, things like tutorials, webinars, publication, including our 40-color publication from last year. And also, you will be seeing there the spectrum viewer when you can build your panel and understand if it's a good combination of fluorochromes. Now, let's focus on extraction or autofluorescence characterization using full spectrum profiling. I'll show you how we do it. And from now, we'll be seeing mainly unstained cells, again, because we want to understand that intrinsic emission of light or the autofluorescence. If I show you lymphocytes and the autofluorescence, this is how they're going to look across all the modules in the instrument and across all the excitation and potential emissions in different wavelengths within each module. What we are seeing in lymphocytes is that they have a discrete intensity, higher emission on UV7, violet 7 detector, a little bit out of the blue, no much emission out of yellow, green, and red detectors, considering this is the 0 in intensity. Now that we can have characterized unstained cells, we can understand how much intensity we are getting, what's the pattern, and how important it's going to be for my assay, based on what I see on my unstained sample, to extract or not that light that's coming from unstained. Let me show you a few other examples, other cell types, how they look. Let's look at red blood cells, tiny elements that don't give you a lot of emission as expected, based on size and complexity. Alveolar macrophages, just from the scatter plot, I can tell they are more granular, more complex. And now that I do a more global characterization of those cells across all the 64 detectors, what we are seeing is the detectors are capturing a lot of light from unstained cells. And the critical point is that now within the plot, the space that I'll have to characterize the real fluorescence from fluorescent dyes, from fluorochromes, is less, and that's where it's so critical to do extraction of this light to have it as your 0. Lymphocytes, we already talked about them, discrete emission. And if we now look at, these are erythroid progenitors derived from umbilical cord in humans. What we are seeing also is that high emission in intensity, high out of the UV, out of the violet, and critical out of the blue. Right here, we see emission where PE is going to be showing you the peak of emission. So critical that you understand that this is going to be different from lymphocytes, that this is what you need to use exactly as the control for your multicolor sample, and also identifying these areas where these cells that are highly autofluorescent in these detectors, areas or detectors in which they don't emit that much light, perfect to characterize the cells and build your panel based on the observations just from unstained cells. Important also, just few examples on how different tissues from the same mice are looking. And what we are comparing here is spleen, kidney, and liver. And you see the differences, right? In the pattern of this emission across the detectors, in the intensity, lower intensity, mid, a little bit higher on the y-axis. We see, for example, out of liver, emission on the red detectors versus not much on the other two tissues. And this emphasizes the need for that perfect match between what you bring as your unstained control and what you have in your multicolor sample. So if you multicolor sample, it's liver, but you do the correction with spleen, you won't be correcting or extracting all the slide that it's in the liver tissue. It's important that we provide you, the software allows you to create multiple controls to have specific correction based on your tissue, based on your cell type. Important consideration, as I mentioned before, is that your staining protocol can modify the amount of autofluorescence you're getting from the cells. These are the same PBMCs, just treated differently. Fresh, fixed with formaldehyde, and permeabilized with FOXP3. What we are seeing is, for this two, more similar pattern, but for this one, changes, especially here in the yellow-green, a lot of yellow-green emission. Important then, that it's not only have a match for your cell type, for your tissue, it's also how you treated the cells, how you treated the multicolor versus how you treated your unstained control to have that perfect extraction based on what you're seeing in unstained cells. And what cool example, and these are algae species, we have 3 microalgae, different species, Synechococcus, Nanochloropsis, and Isochrysis. What we are seeing is that without any exogenous dye, any staining in this microalgae, we are seeing emission of light coming from photosynthetic pigments, from accessory pigments like APC, like PE, naturally occurring in this species. And the important part is now, without adding any staining, you can start characterizing these samples and understanding how things change, how the emission changes when the cyanobacterium are treated, when they are exposed to different microenvironments. It's important that it's not only the autofluorescent helps you not only to be able to extract it, but also to understand your system without any additional stain. And this is a situation that you might have been seeing on your samples already. And as you run your cells from the scatter plot, forward scatter and side scatter, what we are seeing is multiple subsets. When you look at the full characterization of autofluorescence in all our detectors, you are seeing that the pattern is not that clear as we were seeing before. One single box out of each detector. We are seeing multiple profiles with differences in intensity and in the pattern. Well, in that case, what you could do, if we gate on those different subsets on the scatter plot, we, based on defining low, mid, and high populations, and then looking at their profile across the full characterization. If we compare low and mid, looks similar in the pattern. We can tell that mid, it's a little bit brighter on intensity, and definitely the high scatter population, it's showing us a completely different characterization in the ups and downs and intensity as well. But this is visually inspecting the plots. The best way to compare and understand if they are different or not, it's to look at the numbers, look at the median intensities for each detector, and then plotting them together. If we compare the intensity across one single detector, let's say intensity for violet 7 was for this sample 10 to the third, 10 to the fourth, and then 10 to the fifth, clearly changes in intensity, which is what we call changes in brightness. We can also compare across the full signature, compare the spectrum, the ups and downs for this full emission profile, and understand if they are different or not. So if we want to compare, taking into account the brightness, we can plot the median intensity of fluorescence across all those detectors and see clearly here how high that population with high scatter has higher intensity of emission across almost all the detectors when compared to the mid population and the low population. We can also compare the spectrum, the full profile. In order to do that, you want to normalize having the same scale for all these signatures from 0 to 1 in this case, where 1 for each data set will represent the peak of emission. Violet 3, as we see here, for the mid and low, and a completely different peak for the high scatter around violet 7, as we were seeing here as well. Now that I compare on the same scale, I can understand if there are differences in the spectrum. As I see, mid and low are very similar in the spectrum, and the opposite happens with the high scatter, different in multiple areas of that profile that was created, including yellow, green, and red. So now that I know if they are different in brightness, different in spectrum, I can use the software to do the autofluorescence extraction. Let's see how it's done. And there's nothing additional that you need to do. It's letting the software do all the calculation and using the information from your stain to correct and extract that amount of light. What we use for that is spectral unmixing. It's an algorithm that allows you to calculate on the software the contribution of each fluor to your multicolor sample. And that also includes the contribution of your autofluorescence coming using the control from the unstained cells. This is how a multicolor sample will look. It's going to have multiple fluors in there, the autofluorescence. So the software or the algorithm is going to characterize how much light we're getting from the full sample, understand what components are there. And for that, we use single color controls, one control per fluor. So we would use one for the violet dye, one for the blue dye, one for a red dye here, and also unstained cells to know how much light, intrinsic light, is being produced in this system. Once the software is going to do, it's going to start extracting. First, it's going to subtract the autofluorescence, and then going to start deconvolving the signal, removing what's violet, what's red, and what's blue, and finally until the equation equals to 0. After this, you have two options. First option with just regular unmixing is to add back that autofluorescence, or you could let the system extract it. In that case, where extraction is done, what we are doing is defining autofluorescence as another fluor in your panel, characterize it from the unstained cells, and then being able to do the computational removal of it. So let's see how in real data that removal looks like. We have human PBMCs stained with an 18-color panel, and here the scatter profile we want to focus on low scatter cells, lymphocytes. These are unstained lymphocytes. I've shown you this profile before. And let's look at what happened after the staining without using extraction of autofluorescence. After we gated on lymphocytes, we care about the CD3 positives, and this is gated on the CD3 positives. We are looking for CD4s or CD8s, and the plot shows us double negative events and double positive or single positives for each marker. What we are seeing here is that the separation between my negative CD8s and my positive CD8s is not that clear. It's difficult to see the separation or low resolution because all this light, all this signal in the negatives we are seeing, it's coming actually. This is brilliant violet 570 from all this light that unstained cells are giving us in that particular detector, which is the primary detector for 570. So the light's still there, that's the background, and that's what we are seeing. That's why it's high in intensity, 10 to the 4. Similarly, for CD4, high on intensity, and also similar when we gate only on CD8s to understand memory subsets, CCR7, CD45 array. We are seeing how close negatives and positives are in this case, and in the same way for the other marker. Well, if you do one click on the software to do autofluorescence extraction, now what we are seeing is that all this light coming from the unstained sample will be extracted, will be now your zero, and what we are seeing is that the negative population that we were seeing here, close to the positives, is now down to zero, perfect symmetry around zero, and my ability to call what's positive is much, much better because of the better resolution in this plot compared to the top one, and also on this plot compared to what we are seeing, really close populations versus better separated ones. This is an example, but what I want to describe now, it's what you need to do based on how your sample looks, how complex the sample is. You could have different situations or scenarios. You could have a very homogeneous sample, cell lines, sorter cells, where most of your cells are going to have similar brightness, similar spectrum, so extraction can be definitely useful based on what you're seeing on the cells. We will describe examples for each of them. Just give you a general idea. What are the different scenarios? You could have very heterogeneous samples as well, where your tube has more than one population, let's say lymphocytes and monocytes, and then you could have differences in brightness based on how complex and how big those cells are. But it could be the case that even though they are different in brightness, they have similar spectrum. Remember the ups and downs in that signature profile. And the most difficult scenario where you have a very heterogeneous sample, let's say a co-culture of lymphocytes and dendritic cells, lymphocytes and tumor cells, alveolar macrophages, where the cells are not going to be different only based on brightness, but also based on the spectrum. So let's go one by one. Let's start with homogeneous samples, easier to do the extraction. These are induced pluripotent stem cells that were transformed to express GFP, a fluorescent protein. From the scatter plot, what I'm seeing is that it's a very homogeneous population, one single subset in that plot. When we look at those cells in the spectral signature, what we are seeing is that, yeah, we can clearly see the pattern, the intensity, but in general, these rainbow boxes, indicators out of each detector, are really tight, telling me that it's a homogeneous population and that we are dealing with one single autofluorescence signature. Critical now that when the cells were transformed to express GFP, this is how they look. What we are seeing is the GFP, primary detector B1, but below the background, this autofluorescence emission in the cells, even when they don't have any fluorescent protein, that it might make really difficult to distinguish what's the real positivity. So if we go and do unmixing with and without autofluorescence extraction, let's see what we get. For this non-transformed, kind of the unstained control, what we are seeing is there is a lot of background, kind of occupying half of your plot. And this is now the area that we will have to define what's GFP positive. In this GFP expressing ones, if we do unmixing without extraction, what we see is the high background of the negatives, our positives really dim, as we were seeing before, and issues with identifying or separating positives from negatives. If we now do unmixing with autofluorescence extraction, all these lights from the negatives will be extracted. That's going to be your zero. And the positives will stay at the same intensity, but now my ability to separate these positives, gate on them, it's clearly much, much better and better estimation of the positivity when extraction is done, because this negative or this non-transfected, this background was extracted, considering the background as a new fluorochrome, new autofluorescence tag in your panel. One more example, this time without showing you UV illumination, because you still can do it on other configuration if you don't, even if you don't have UV modules or UV laser. These are HeLa cells, again, very homogeneous. The spread of the data, the CVs for these rainbow boxes are really tight, which indicates it's homogeneous sample you're dealing with. High background from the non-transform in the critical detectors for your M-cherry expression. And again, that might difficult to understand what's the positivity. Non-transformed, high background on M-cherry expression, the unmixed for M-cherry. We are seeing the positives, but the high background makes difficult to know where to gate. Versus the unmixing with autofluorescence extraction. We can now see the negatives down to 0, the light was extracted, and the positives clearly separated, where we can define percentage of positive cells for that M-cherry expression clearly compared to without extracting the autofluorescence. So extremely critical and easy if you have a homogeneous population. If you have a heterogeneous, now it's going to depend on how different those cells are in brightness and spectrum. The case for PBMCs, peripheral blood mononuclear cells, is they tend to be showing you differences in brightness but similar spectrum. Let's look at the scatter profile, lymphocytes and monocytes. If we look at all the events, this is the signature we get. This time, the boxes are not that tight. This time, what we are getting, it's kind of 2 boxes on each single detector, reflecting the differences in intensity between the low box, the lymphocytes, and the brighter box, the monocytes. What we get from here is that if we gate only on monocytes or lymphocytes to show the profile, now it's very tight. It's clearly distinct for them. The pattern looks similar across all detectors, and the intensity is dimmer on lymphocytes, brighter in monocytes. But visually, it's difficult to conclude. The best way to do it is exporting the medians for each detector and comparing. We have a tool provided to you to do that comparison. If we compare based on brightness, based on the median intensity of fluorescence, we see lymphocytes on orange, dimmer, and on blue, monocytes brighter across multiple detectors. If now we use the same scale for both, taking brightness out of the equation, comparing only the spectrum, we see that they are highly overlapping. Very similar ups and downs in those signatures that are going to be reflecting on their results. And let me show you for the same 18 color panel, those 2 subsets. What happens if I do unmixing with extraction using one or the other one as your new tag, new fluorochrome in your panel? If you use all the events, or if you use lymphocytes on monocytes, and here just showing you one plot, CD4 versus CCR7, a plot where we should be seeing double negatives, double positives, and single positives for each marker, and the same plot for each condition. If we compare across the 3 conditions where different subsets were used for your autofluorescence extraction, there are no differences because their spectrum was similar. Then the unmixed data should be similar. The other case would be where your populations are so different in the spectrum that you need to do specific corrections or add additional fluorochromes, not only the one that comes with the software, but adding more. And you can add as many as needed based on how complex and heterogeneous your sample is. In this case, what I'm going to do is show you an example. What went wrong with one experiment and show you how the approach, understanding that the sample was heterogeneous and then doing the multiple extraction helped to get better resolution in the data. This is an 8-color panel. These are bone marrow cells in mouse. And what we did initially was just autofluorescence extraction. We have multiple subsets. What we did was eliminate unwanted events. We remove cell debris. We remove all these marginal bright events. And what we are getting here is the single cells. We remove any cell doublets and aggregates. And we also remove dead cells using a viability diet. And we pay close attention to the stability of the fluorescent signal using time as a parameter. After that, using a sample where the multiple single color controls were merged, what we are seeing is expecting to see is the negatives in alignment with the positives for each marker and multiple permutations shown here. So we see that alignment. We know there were no issues with the unmixing or the spillover correction. What we are also seeing is this diagonal, again, not explained by unmixing issues, not explained by having debris in the sample, not explained by having dead cells. If we compare the same plots in this multicolor sample with what we see in the unstain, we now understand where that diagonal or those different diagonals are coming from. In the unstain, we see most of the events low on intensity for those fluors, but we see false positives. In the diagonals have very similar shape when we compare top and bottom, indicating there is an additional population in this unstained control that has a very different profile of autofluorescence that is giving us these false positives on multiple dyes and that needs to be extracted independently based on its own profile. So having those diagonals where they are not explained by any issues with your unmixing or by having any antibody aggregates, cell aggregates, or dead cells, it's clearly an indication that autofluorescence is playing a role. And the good thing is, since you have an unstained sample, you can understand what happened and create a particular signature just for this population and then characterize it and extract it. So how do I know I have a high degree of heterogeneity in my sample? Well, let's start with the scatter plot. From here, I know I have 1, 2, 3 populations, and I can look at their signature profiles. I was able to separate up to 4 populations. If we look at the low scatter, mid, and part of that high scatter, and if we look at the signatures, they look similar. Go up on UV, up on violets, in a similar pattern. But there were some events in that high scatter that are very different. Since we have multiple detectors, we have multiple opportunities to understand and characterize those events, understand they are different, and then use those differences to do specific extraction. Our suggestion, if this is your case, if when you explore the sample, this is what you're seeing, is now you can define an additional autofluorescence signature using these events and include it in your panel. How would you do that? We always go to the same way to compare the signatures, independently if it's homogeneous, heterogeneous, and differences in the spectrum of brightness. The way to recognize those differences is by plotting them together. So that's what we did. The 4 signatures were plotted together to understand their median intensity of fluorescence, and we see clearly how the high scatter population is so different in intensity, brighter than others. When now I compare the normalized signature, 0 to 1, same scale, I can see how the 3 that are low on scatter are similar, overlapping in emission, and something that is completely different, that's giving me a different spectrum, and that now I can consider as a new autofluorescent tag, new fluorochrome, since I have the unstained cells to define that new fluorochrome. You don't really need to go and do all that exploration on your own, looking at the signatures. There is a way to do it on the software. On the software, you can calculate a metric called similarity index. Cytek defined this metric to understand how similar 2 signatures are. The similarity index goes from 0, no similarity, to 1, identical, and that number can be calculated on the software. So if we compare low, mid, the high, low, and the high, high, what we're seeing is the numbers for the 3 initial ones are really close to 1, indicating that they are similar, low versus mid, close to 1, mid versus high, low, also close to 1. That similarity, it's on something that we had already concluded from here. These 3 signatures look very similar, but the software can make that conclusion for you. What we are also seeing is that the high signature, it's different from the others. And with, as you would do with fluorochromes, similarity of 0.98 or less than that tells you that that signature is unique from the other autofluorescence, unique also from your fluorochromes, and needs to be extracted independently. What you do next? That autofluorescence that is defined as different can be included in the panel as a new fluorochrome. We can then understand how similar it is to other fluorochromes in there. As we see intermediate similarity with PERCP, now it will be corrected properly. And in order to do that, similar to any fluorochrome, similar to what you would do in conventional, having single color controls, to train the software and understanding what's FITC, what's PE, what are the different fluors present. Since now my autofluorescence will be a new fluor, I need to have a way to understand what that fluor looks like. For that, you will need to define a new control, defining positives, the signature we just identified, defining negatives, the events with lowest emission, and then adding that to your experiment. If we talk about the control, the rules for those single color controls or reference controls, the same rules will apply and similar to the rules that you would use for conventional cytometry. Your control needs to be as bright or brighter than your actual sample. The spectrum in that control needs to match what you have in the multicolor sample. So now the match is going to be same cells, same preparation in your unstain and in your multicolor. Another important consideration is that you have enough events, so to acquire a lot of events in your unstain to do this characterization properly. And the last rule is that the negative and the positive should have similar autofluorescence. It's not possible in this case. We are starting from the point that they are actually different. So we will define that rule and use that signature as your positives. And for the negatives, use the lowest autofluorescence as possible. Once you do that, you define the new one in your software. The software will do everything for you. All the calculation, all the correction, all the extraction. And what we will get at the end, if we go back to that example where I'm mixing with autofluorescence extraction, what extraction for the lymphocytes or the low scatter population was given as that diagonal, is that now that I know what diagonal it is coming from, the height scatter, I add that height scatter to my experiment. I extract it. You see how that's impacting the quality of the data and definitely eliminating those false positives from every single permutation that is plotted. But if we now look at their data, how that was impacting their ability to see their populations of interest. If we go from unmixing and multiple plots, but key points are, they needed to define CD19 double positives. There is some diagonals here. The negatives are high in intensity, and it's difficult to separate what's positive and what's negative. For all the other plots, independently of the marker, what we are seeing that it seems like they are double positive. They are in that upper right quadrant indicating double positivity. But when we started using and characterizing autofluorescence and extracting separately, by defining multiple tags, what we see now, and if we compare top with bottom, is that negatives are down to 0. These diagonals are eliminated. My ability to define positivity for CD19, it's much, much better, clear separation. And now for the other markers, I see now what's expected. CD23, for example, a subset out of the B220 population, and CD23 a subset out of the IgM positive population. These two subsets that were dim in intensity, that were really close to the negatives, were not identified initially when the autofluorescence was not extracted. That autofluorescence was still dead. The background was masking these positive signals. And now doing extraction clearly reveals the true fluorescence of these dimly expressed markers. The benefits of autofluorescence across multiple samples are really clear, giving you in general better resolution for your data. And on this particular part, the critical point is that you can define, if your sample is very heterogeneous, you can define multiple tags to characterize the different subsets in there out of your unstained sample and do the correction to get better resolution. To conclude, full spectrum profiling gives us the ability to characterize and extract autofluorescence if needed in order to improve the resolution in your data. We have seen how the impact of the autofluorescence is going to be different based on what cells you have, how they are treated, and what are the staining protocol. So make sure that if you want to characterize well the autofluorescence, you bring unstained samples with enough events to characterize it better, that unstained sample matches the sample type and the preparation method you use in your multicolor, And then you define what's the optimal unmixing strategy by looking at how much light you're getting, how heterogeneous is, and how many subsets you are present in your sample. Just letting you know, and you can use this code to get extra information in our newsletter. We're going to have more webinars focused on autofluorescence, discussing difficult cases with not only one autofluorescence signature, but multiple ones were needed to do a good extraction. When we will be showing you how critical is now that you understand the autofluorescence in your cells, then how you design panel around that, what fluors are going to be better, extraction needed, and the separation for your different fluors and the resolution. And finally, some examples on how we can use autofluorescence as a label-free method to conclude without any exogenous agents. Well, that's the end of my presentation. Thank you so much for your attention. And I'll be happy to answer any questions. Let me say thank you to a fantastic team at Cytek Biosciences, our customers that provide data and sets. And thank you so much. Be happy to answer any questions you might have.
Thank you, Diana, for your informative presentation. We're now going to start the live Q&A portion of the webinar. So if you have any questions you'd like to ask, please click on the Ask a Question box located on the far left of your screen, and we're going to answer as many questions as we have time for. So let's get started. Our first question is about a project on aging, and they're wondering, based on your experience, will aging autofluorescence be a problem in FACS?
Well, for aging, there are two studies that I know. One is on, and I'm telling you this because it's both studies had the same conclusion in terms of what protein was involved when evaluating autofluorescence, no by flow cytometry, microscopy, or imaging. So one of them is using C. elegans, and when C. elegans is aging, it produces more lipofuscin, which is one of those components within the cells that can emit light, red light mainly. So that would be an indication that it could work. That finding, I know it's a different system, but also in humans, when you do imaging of the eye as well, when there is macular degeneration, you can also see increased levels of lipofuscin. So that could be an indication that you can do the characterization upon aging. But also there is a mouse study in brain characterizing autofluorescence in microglia. And one of the conclusions is that cells or cells from mouse systems that are aging, they see higher autofluorescence. This time not doing any specific characterization on what component it is, but definitely there are suggestions in the literature that aging, it's going to be increasing autofluorescence, and in particular through at least C. elegans and humans through that lipofuscin pigment, which is just a lipopigment.
Let's go to the next question. This one's related to tumor and clinical samples where you may not have lots of sample to do an unseen control. They're asking, Diana, in your experience, what is the minimum number of cells needed to be collected for the autofluorescence extraction to work properly?
Yeah, that's a difficult point. It's going to depend on how frequent your cells with a different autofluorescence profile are, what's their frequency mainly. So since we are defining autofluorescence as a new fluorescent tag, it's treated as any fluorochrome or any reference control in your experiment, then the guides will be the same. It's 500 minimum. 500 cells or events minimum to characterize it. But what I've seen so far is that the more, the better. The more cells you acquire, and I know it can be difficult in that scenario where tumors, where you don't have a lot of cellularity, but the best has been so far where you have a sample where many events have been recorded, 100,000 for that unstained control, in order to capture those populations with low frequency that are the ones that have different autofluorescence profile. I would do it at least the first time if it's a kind of a longitudinal study with all your samples, so you understand better your system, your particular tissue, your particular conditions, and if it's a tumor, your infiltrate or the actual stromal cells. But yeah, a critical point, having very few makes difficult to understand the signature. Having very few then makes difficult to do the extraction in a representative population of what you will have in that multicolor sample with more and more events.
Okay, thanks, Diana. Let's go. Let's try and answer as many as we can. So the next one is the next question is about a panel where they're using more than 20 colors and unmixing doesn't actually work like we showed. Then they're having to do a lot of post-unmixing compensation. Is there a possibility to improve the unmixing results?
Oh, yeah, we can, if you know the name of the technical application specialist in your area, we can help you review. What we have discussed here it's not related to the fluorescence controls or the reference controls and the unmixing calculation to correct for the spillover between fluors. It's mainly unstained cells and how we treat that information. So if there are issues with your unmixing calculation for the fluorescent dyes, it might be reflecting a problem with your reference control, that control for your FITC, for your PE, and not an autofluorescence. In general, if we see diagonals, it's either a mixing error because of your control or autofluorescence. So two separate things. One, we use the unstained sample for autofluorescence to characterize and then do that extraction. Multiple extractions if needed. The other one needs to be addressed from the reference controls, the quality of the controls, the type of control you are using, cells versus beads, and how that's useful for the unmixing of fluorochromes. So my suggestion is to contact your application specialist. Let us know in the chat if you know the name of that person, and so they can help you understand better on your data set what's the situation and how that can be fixed.
Thank you. The next question, can you create an autofluorescence tag from data that's already been acquired? And kind of on a similar note, there's a separate question, is autofluorescence extraction done post-analysis, or can we see the results live?
Sure, we can do it live because it's once you record your unstained sample, your reference controls, you are able to do the unmixing, and so that's live. You can acquire your sample live after doing the calculation, after doing the extraction. It doesn't need to be done after acquisition, but both options are available.
Okay, great. And let's see, maybe one or two more questions, depending on. So the next one, is it possible to over extract your autofluorescence from a mixed population? For example, on lymphocytes, if you have a lot of cells with higher autofluorescence than your population of interest.
Yeah, our recommendation is to try both options, with and without extraction, because there are some cases where the autofluorescence is not really high, it's low, that the correction, since it's based on the median from the unstained, will give you unexpected results or more spread in the negatives. So yeah, it can be detrimental, that extraction, and it's a good point. For most of the cells, especially if you have big cells, granular cells, it's going to be useful. But we have seen cases in which that extraction lead to less resolution. So ideally, you can create two experiments, without extraction, with extraction, and compare. Look at the resolution, look at your populations, and from the biological point, see if the results make sense.
Okay, and let's go to the next one. How do you define the detectors needed to create the autofluorescence tag?
Good point. Basically, from that evaluation that we do, looking at the signature profile, where you see, and if you see a very heterogeneous sample, you could do one is check all the permutations and see where populations are separated, just from the autofluorescence. That's one option. The other one is use that signature to guide your selection or your definition of what detectors to select. And by that, I mean, if in the signature you see that there are kind of two different profiles, and one of the detectors allows you to see them clearly, the separation, that could be an ideal one. The other approach could be gating if the scatter plot is different, gating on the scatter populations, and then looking at their single profiles separated and defining what are the detectors that allow me to define the differences in order to identify that population that will help you to define negatives and positives, or at least positives. But if the selection of the detectors can be done in multiple ways, as I described, but it's mainly based on what you are seeing on the signature in terms of intensity and differences across the same detector for the different populations.
Okay, I think we're out of time. Thank you again, Diana, for your time and your insights into autofluorescence. I'd also like to thank LabRoots for hosting this. Before we go, thank you all for joining us today and for your interesting questions. Any questions that were not answered today by the speaker will be addressed via the contact information you provided when you registered. The webcast can also be viewed on demand, so LabRoots will be sending you an email when it's available for replay, and we encourage you to share that email with all of your colleagues who may have missed today's live webinar. Thank you again. Bye-bye.
Bye.